Thank you in advance for that applause. Thank you to OSI, and congratulations on your 10-year anniversary. We are a public company, so I do need to put up our forward-looking statement. Our objective is to build a leading retinal company, and to do that, we're going to develop transformative gene therapies and novel therapeutics for retinal diseases. Our focus will be on large and orphan indications. We've built our pipeline through business development, and we'll continue to do that through business development, focusing on really three key criteria: innovative medicine and science, unmet medical need, and a viable commercial opportunity. Our execution has been predicated on a strong team of 35 people that I've worked with over the last year and a half. We'll talk a little bit more about them. A lot of experience in doing clinical trials, and we'll talk about their success over the last year. And obviously, we have a strong cash position to drive future growth. We've taken a multimodal approach to building or maximizing our potential for success. We have two categories: therapeutics, where we have a deep understanding and expertise in this field, and a global network of KOLs and clinical sites that we can tap on to do those trials. And we've added gene therapy over the last 12 months to our portfolio of pipeline, focusing on novel and cutting-edge AAV therapies. Just to take a look at the pipeline as it stands today, I presented to you last year, and this was a chart that had 2 clinical programs, 3 active programs. I'm happy to say today there's 8 programs on here, and hopefully more to come. I'll briefly cover the summary here on the therapeutics and gene therapy and go into a little bit more detail in the coming slides. But on the wet program, our phase 2A program that is ongoing, that's in combination with anti-VEGF therapy, Lucentis. We have a GA trial secondary to dry AMD. That's a 2B trial. That is a monotherapy trial and expected to have data next year. We have a Stargardt's trial ongoing. That's a 2B trial as well. And we also have an IPCV, which is a degenerative retinal disease largely prevalent in Asian population. That's in its early days. But we're happy to report our initiatives in gene therapy as well. We recently signed a collaboration with the University of Pennsylvania and University of Florida for RHO-ADRP. I'll talk about that a little bit more. That's a program that has an opportunity next year to do IND-enabling studies and hopefully move into the clinic by 2020. And we have 3 programs that we initiated with the University of Massachusetts Medical School on novel gene delivery technologies, LCA10 using their Minigene, and the Stargardt disease using also their Minigene. A little bit about each of the programs. Our wet trial using Zimura in combination with anti-VEGF. This remains a major market opportunity with unmet medical need. Anti-VEGF therapy has been very effective here, but it's reached the ceiling effect. And in real-world situations, patients lose vision and return to baseline and below, and many of them develop geographic atrophy. The role of anti-VEGF does activate complement, and we believe by adding Zimura to anti-VEGF, we may have an opportunity to improve efficacy and safety for patients. This is not the first time that we had Zimura in patients. We previously finished a 1A2 trial, small uncontrolled safety trial, in which the inclusion criteria were naive patients, all CMV subtypes, receiving 6 doses of Zimura with Lucentis over a 6-month period. The safety profile was good. All doses were well tolerated, and there were no safety concerns. The important data is to the right, and again, I caution that this was an uncontrolled study, but if you look at the dose escalation here, you see improvements in 3-line gain from 46, 47, and 60%, which was quite compelling. If you look at today's treatment with anti-VEGF only, those numbers are normally in the 30 to 40% range. This was compelling enough for us to initiate a 2A open-label clinical trial with 64 patients. That trial is fully enrolled now. The objectives are to assess the safety of intravitreal Zimura with Lucentis. There is a dose-ranging element to the study as well, and we're hoping to validate the phase 1A2 results and move this program forward. Duration of treatment, 6 months, and we're hoping to have data before the end of the year. Our second therapeutic trial is geographic atrophy secondary to dry AMD. Again, a large market opportunity, unmet medical need. There's probably 1 million patients here in the United States, 5 million worldwide, and there are no approved treatments for this terrible disease. We believe there is a genetic link between complement and AMD, and with aging, we see the buildup of inflammasomes and MAC attack complex, both when they form, present problems with retinal pigmentation and lead to cell death. Our program designed here is a phase 2B program, which is randomized, double-masked, sham-controlled, having 3 cohorts of drug, 1 of sham. We've enrolled and have completed 286 patients into this trial. The study had a monthly treatment of Zimura, and we'll track these patients over an 18-month period. The primary efficacy endpoint here will be mean rate of change in GA over a 12-month period, measured by fundus autofluorescence at 3 different time points. Expect data fourth quarter of next year. Last trial I'll talk about on the therapeutic side is the Stargardt trial. Again, an area of unmet medical need. It is an orphan disease. There are about 62,000 to 77,000 patients in the US and Europe that suffer. As you know, many of these are younger patients. This is a progressive disease that targets the macula and the retina, and it is caused by a mutation in the ABCA4 gene. We have a current trial ongoing here. It's a phase 2B trial. It's a randomized, double-masked, sham-controlled trial. There are 2 arms, Zimura and sham. We expect to enroll up to 120 patients. The primary endpoint here would be change in the area of the ellipsoid zone, defect measured by ONFOS SD-OCT. Top-line data, 2020. We're also happy to be working with the Foundation Fighting Blindness that has given us access to their publicly available ProgStar data and also has helped in a patient registry and facilitating enrollment in this trial. So a little bit about gene therapy, which is new to us over the last year. Why, if we are going to be a leading retinal company, we felt it was essential to add gene therapy to our portfolio. Many ocular diseases with indications are due to genetic defects. The potential to cure disease is quite compelling in any area, whether it's ophthalmology or otherwise. But the eye is very conducive to gene therapy. And obviously, if anybody in this room and us are successful, tremendous positive implications for patients and caregivers. The first program, our lead program on rhodopsin-mediated autosomal dominant retinitis pigmentosus. This is the most prevalent inherited retinal disease. And I point out on the bottom, there's more than 150 identified mutations of RHO. Our program looks to, it's a mutation-independent strategy with a single AAV vector technology that looks to silence out and knock out mutated toxic rhodopsin proteins by producing a healthy wild-type rhodopsin protein. This was tested in animal models at UPenn, and there was a paper that was published in August covering that, and we hope that we see a clear path to IND in 2019 and enter the clinic in 2020. The second area of gene therapy is our collaboration with the University of Massachusetts, working on vector technology using their Minigene concept. Very simply, it's taking a shortened version of a functional gene that was a larger gene and making it fit in the existing AAV technology, which is both well-tested and very safe. As you know, the capacity of the current vectors are limited to 5 kb. The Minigene would hopefully design that gene in a way that would fit efficiently. We are working on 2 programs using the Minigene technology. The first is Leber Congenital Amaurosis, focusing on the CEP290 mutation, which leads to early onset of vision loss. And the second, Stargardt disease, focused on the ABCA4 gene. And again, as I talked about before, this is a progressive and disease that damages both the macula and the retina. The third program that we have with them is in the area of novel gene delivery, and more to report on that as we move forward. So in summary, happy to be here. I think we've made tremendous progress in building the portfolio, focusing on retinal diseases. We have a number of data points that are coming up. Wet data later this year, dry data in Zimura in 2019, Stargardt data in 2020, all Zimura programs. And on the RHO ADRP preclinical program, we'll start our IND-enabling studies in 2019 and hopefully reach the clinic in 2020. Our strategy to build the portfolio has been business development. We'll continue to do that. We continue to look and expect to add to the portfolio. And finally, we have a strong cash position to drive this growth with $146 million at the end of the second quarter. Thank you.